Today I'll be reviewing the general points of the Lac Operon. It is found in bacteria and is needed to metabolize certain food sources. While the main energy sources are glucose and lactose, glucose is always preferred. Let's take a closer look. Under the right conditions, the beta-gal gene can be turned on to produce the enzyme beta-gal. The function of beta-gal is to metabolize lactose into a simpler form. So we know that we need the presence of lactose to turn on the beta-gal gene, but what else is necessary? There are two other players or binding sites that are important to this process. The rep binding site is sensitive to lactose. Rep will bind to the rep binding site only when there is no lactose present. This is how it represses the system. When this occurs, no beta-gal will be produced. If there was lactose present, then it'll bind to rep, leaving the site unbound. An unbound rep binding site is part of what we need to produce the beta-gal. But there is also another binding site. This one is sensitive to glucose, and it's a little bit more complicated. If glucose is present, then it'll block the molecule CAMP from binding to CAP. Plain CAP will not bind to the CAP binding site. Without glucose present, CAMP will be produced, it binds to CAP, and this goes on to bind to the CAP binding site. In order to make the final protein beta-gal, we need CAP to be bound. So what combination of these two will induce or inhibit production of beta-gal? First, let's look at how we get beta-gal to be turned on. We need lactose to be present, resulting in an empty rep binding site. And we also need glucose to be absent in order to get binding of the CAP binding site. If these two conditions occur, the circuit is considered on, and we will produce the beta-gal enzyme. But remember, both binding sites are separate and they need to be treated that way. Now, which conditions would block production? There must be no lactose present, and there must be glucose available. Let's review all the possible conditions. Keep in mind the top left corner shows how we turn the circuit on. If both sources are present, neither CAP nor rep will bind. The beta-gal gene will not be transcribed. If glucose is present but lactose is absent, again we see no beta-gal production. When only lactose is present, we do see transcription of the beta-gal gene. Notice that the conditions match the top left corner. And when neither are present, no beta-gal will be made. To summarize, we only need to turn on transcription of the beta-gal gene when we have lactose present with no glucose. Doo-doo!